Gas exchange in the human body is a vital process that ensures the proper functioning of our cells and organs. It primarily occurs in the lungs, where oxygen from the air is taken in and carbon dioxide, a waste product, is removed. When we breathe in, air enters the respiratory system through the nose or mouth and travels down the trachea. The trachea branches into smaller tubes called bronchi, which further divide into even smaller tubes called bronchioles. At the end of these bronchioles are tiny air sacs called alveoli. The alveoli are the key players in gas exchange. They are surrounded by a network of blood vessels called capillaries. Oxygen from the inhaled air diffuses across the thin walls of the alveoli and into the capillaries, where it binds to red blood cells and is transported throughout the body. At the same time, carbon dioxide, produced as a waste product by our cells, diffuses from the capillaries into the alveoli and is then exhaled. This exchange of gases is made possible by the difference in their concentrations between the air in the alveoli and the blood in the capillaries. The process is facilitated by the large surface area of the alveoli, their thin walls, and the constant flow of blood through the capillaries. Overall, gas exchange in the human body is a remarkable and essential process that allows us to obtain the oxygen we need for energy production and remove the waste product, carbon dioxide, from our system. CO2 is dissolved in plasma through a process called simple diffusion. When CO2 is produced as a waste product in our cells, it diffuses across the cell membranes and into the bloodstream. CO2 molecules are small and nonpolar, which allows them to easily pass through the lipid bilayer of plasma membranes. Once in the plasma, CO2 molecules dissolve directly into the aqueous solution. The solubility of CO2 in plasma is influenced by several factors, such as temperature, pH, and partial pressure of CO2. Higher temperatures and lower pH levels increase the solubility of CO2 in plasma. Additionally, the partial pressure of CO2 in the blood, which is determined by the rate of CO2 production and elimination, also affects the amount of CO2 that can dissolve in plasma. The binding of CO2 to hemoglobin occurs in the peripheral tissues where carbon dioxide is produced as a waste product of cellular metabolism. Deoxygenated hemoglobin, Hb, in the red blood cells undergoes a conformational change that promotes the binding of the released hydrogen ions, H+. The binding of the hydrogen ions to hemoglobin promotes the release of oxygen molecules from hemoglobin, allowing it to be delivered to the peripheral tissues. As oxygen is released, hemoglobin can bind with carbon dioxide to form carbaminohemoglobin. CO2 binds to the globin chains of hemoglobin, mainly to the amino acids present in the N-terminal region. The binding of carbon dioxide to hemoglobin helps in its transportation from the peripheral tissues back to the lungs. When the blood reaches the lungs, the opposite reactions occur. The high concentration of oxygen in the lungs promotes the release of carbon dioxide from carbaminohemoglobin. The released CO2 diffuses into the alveoli of the lungs and is ultimately exhaled during the process of respiration. Carbon dioxide, CO2, is transported in the blood primarily as bicarbonate ions, HCO3. The majority of CO2 molecules produced in the tissues are converted into bicarbonate ions before being transported to the lungs for elimination. The process of CO2 transportation using bicarbonate ions is known as the bicarbonate buffer system. In the peripheral tissues, CO2 diffuses into the red blood cells, RBCs, due to its high concentration in the tissues. Once inside the RBCs, CO2 combines with water, H2O, to form carbonic acid, H2CO3, in the presence of an enzyme called carbonic anhydrase. This reaction occurs rapidly and reversibly. Carbonic acid, H2CO3, then dissociates into bicarbonate ions, HCO3, and hydrogen ions, H+. Bicarbonate ions, HCO3, are negatively charged and can exit the RBCs in exchange for chloride ions, Cl, through a chloride shift mechanism, maintaining electrical neutrality. This allows the bicarbonate ions to enter the bloodstream. The majority of bicarbonate ions produced in the RBCs are transported in the plasma, dissolved in the blood, while some remain within the RBCs. In the lungs, where the concentration of CO2 is lower, the reaction reverses. Bicarbonate ions, HCO3, re-enter the RBCs in exchange for chloride ions, Cl, through the chloride shift mechanism. Carbonic anhydrase enzyme present in the RBCs catalyzes the conversion of carbonic acid, H2CO3, back into CO2 and water, H2O. The CO2 molecules are then transported out of the RBCs and into the alveoli of the lungs, where they can be exhaled during respiration. The Haldane effect is a phenomenon observed in the field of respiratory physiology that describes the relationship between oxygen and carbon dioxide transport in the blood. In simple terms, the Haldane effect states that the affinity of hemoglobin for carbon dioxide, CO2, is inversely related to its affinity for oxygen, O2. This means that when hemoglobin binds with oxygen, its ability to carry carbon dioxide decreases, and vice versa. This concept is closely related to the binding mechanism of oxygen and carbon dioxide in the blood. When oxygen-rich blood enters the lungs, the high concentration of oxygen causes hemoglobin to bind with oxygen molecules. This oxygenation of hemoglobin promotes the release of carbon dioxide molecules from the hemoglobin, which then diffuse across the alveolar capillary membrane and are expelled from the body during exhalation. In contrast, in tissues with lower oxygen concentration, such as active muscles, hemoglobin releases the bound oxygen molecules and readily binds with carbon dioxide, facilitating its transport back to the lungs. This mechanism allows for efficient removal of carbon dioxide from tissues and its subsequent elimination from the body. The Haldane effect also explains the role of carbon dioxide in maintaining blood pH. As carbon dioxide is transported in the blood, it undergoes a chemical reaction with water, forming carbonic acid. This reaction is catalyzed by the enzyme carbonic anhydrase. Carbonic acid then dissociates into hydrogen ions, H+, and bicarbonate ions, HCO3. The release of carbon dioxide by hemoglobin promotes the formation of bicarbonate ions, which serve as a buffer to maintain the blood's pH within a narrow range.